Did you know that 1% of Canadians are infected with the hepatitis C virus? Also known as HCV for short, this virus causes chronic infections that can lead to life-threatening complications, including liver cirrhosis and liver cancer, and it's the leading cause of liver transplant in our country. This makes HCV a major public health concern. The thing that I'm most interested in regarding HCV, however, is its unusual interaction with miR-122, a microRNA specifically expressed in the liver. If you don't know what microRNAs do, they are small RNAs that bind to the 3' end of target messenger RNAs, and this represses translation from the target mRNA and promotes its decay, thereby regulating gene expression in this way. At least that's in the normal cases. But I told you that HCV has an uncharacteristic interaction with miR-122, right? As it turns out, the HCV genome recruits two molecules of miR-122 to its 5' end. And instead of downregulating translation, this interaction actually promotes HCV translation and RNA accumulation. But how does that work? Well, recent studies have shown that miR-122 binding promotes the HCV life cycle by fulfilling at least three distinct functions. Depicted on my slide is just the 5' end of the viral genome where miR-122 binds. miR-122 first acts as a riboswitch, allowing the viral internal ribosome entry site to form, which is the structure that permits kappa-independent translation. miR-122 also stabilizes the viral RNA by protecting its 5' terminus from exoribonucleases, and it promotes translation. Now, these three roles are pretty intimately linked, but we're interested in figuring out which of the three contributes the most to the overall impact of miR-122 on the HCV lifecycle. By using different mutants of HCV, we are able, for the first time, to isolate these three roles and assess their impact on HCV translation and RNA accumulation individually. Using luciferase assay as an output for translation and qPCR to measure viral RNA levels, I'm currently quantifying the difference in viral dynamics when individual roles are removed and fulfilled. Thus far, our results seem to indicate that translational promotion seems to be the dominant of these three roles, while riboswitch has the lowest impact. By detailing the mechanism of action of miR-122 in promoting the HCV lifecycle, I'm hoping to shed more light into this unusual phenomenon of a microRNA that promotes RNA stability and translation as opposed to repressing them. Similar non-canonical microRNA interactions have been observed both in cellular systems and in other viruses, and we are only beginning to understand the multifaceted life of these small RNAs. Therefore, my research has the potential to provide insight into these unusual interactions and how we can target them to treat viral infections or study cellular processes.